Welcome back, friends. So we are talking about recombinant DNA technology, and we have discussed about the cloning vectors. Now in this video we'll be talking about expression vectors. Now expression vectors are different than the cloning vectors in several different ways, because the cloning vectors, remember what are vectors actually? Vectors are vehicles for transferring our desired gene from one location to another location. That's right? We know that. Now a cloning vector is that, so suppose this is our gene of interest, and cloning vector is our vehicle of transferring this gene of interest into our host cell. So if this is the vector, so we'll take this and attach this to our vector. We call it a recombinant DNA molecule now because it is carrying our insert. Now we will transfer it into the host cell, which can be bacterial cell or which can be eukaryotic system. But this is the job of a cloning vector. So job of a cloning vector ends when it enters into a host cell, but the job of a expression vector begins when it enters into the host cell. You can look at it in this way, right? So when this vector insert itself into this host cell, suppose this is the host cell, and inside this host cell we are having this recombinant molecule. So here this one, let's consider this one. So this is our recombinant molecule. It enters. So the job of this cloning vector, which is in red color, is done now, but the job of expression vector will begin once it is transferred into the host cell, because as the name suggests, expression vector, the utilization of this vector is for the expression of our desired gene, not the transfer of our desired gene. Cloning vector deals with the transfer of our desired gene and screening of our desired gene from the library, but expression vector deals with the expression of that desired gene. That means we need to make mRNA from there, and we need to make proteins by translating this mRNA, right? So we need to make this protein inside the cell, and we can achieve this by the expression vector. So that is the difference factor about expression vector and cloning vector. So what is expression vector? An expression vector, it is also known as expression construct, is a plasmid or virus designed for the protein expression inside the cell. So it's designed for the protein expression. That's the actual part. Now what are the goal of this expression vector? The goal is the production of significant amount of stable messenger RNA. That means to produce significant amount of proteins, right? So this is our actual goal, to produce significant amount of proteins. That means the expression of our desired gene, right? Because if we know the central dogma of biology, gene of interest, which is DNA, from there it will produce mRNA, which is the messenger, then it to produce proteins. This is the central dogma. So this part is called the expression, and we are dealing with this expression during this expression vectors. Now what are the elements of expression vector? Remember, I have told you before, when you talked about that cloning vectors, that there are some certain criteria that must be fulfilled by a DNA sequence to be cloning vector. They must have some important sequences in them. The sequences are three important sequences. One is the origin of replication, second one is the multiple cloning site, and also selectable markers. But in this case what we will be looking, we will be looking that for a DNA sequence to be expression vector, it must have all those three features which is carried by the cloning vectors. So it must have all the features that are there in cloning vector, but it must have certain more features, right? So this type of expression vector is having much more elements than a cloning vector. It is having that three elements which are common to all kinds of vector, but instead of those things, it have this five different type of more important features. One is that it must have a strong promoter, and this promoter must be inducible. So it must have a strong inducible promoter. Now what we mean by inducible promoter? Inducible promoter means is that kind of promoter which can be induced in certain conditions. In normal situations, the promoter, so suppose for understanding of this expression vector, you must understand the Operon concept very well. So if you don't know Operon concept, just go back in my YouTube channel and you will find videos of Operon. You will find the videos of lac Operon. Just go through that video, then come back here. It will help you to understand. Now say in Operon concept, what we know, that there must be a promoter region and operator region right beside that. Then we are having a desired gene of interest. So this one is the desired gene of interest. Now what is going on? Inducible promoter means those type of promoters which can be influenced, which can be induced by the presence of certain chemicals like IPTG, right? That means we can control the expression of that particular gene if we regulate the binding of certain inducers to the promoter. So normally the promoter will express very little amount of the gene, but when we add the inducer like IPTG, it will dramatically increase the expression of the desired gene, right? So it must be inducible promoter, and it must be a strong promoter. So what we mean by a strong promoter, a strong promoter always bind with its sequence, or a strong promoter is very well-known promoter for binding with the RNA polymerase, which is the transcribing machine, most important transcribing machine of all. So RNA polymerase is the enzyme responsible for production of mRNA from the DNA. So what it can do, strong promoter means it is having higher affinity towards the uh, RNA polymerase. Okay. Second thing is that it must have this translational initiation sequence, because we are destined for the expression purpose. So we must have transcription and translational machinery initiation and termination factors. So it must have the ribosomal binding site, obviously a start codon, and also termination sequence, transcription termination sequence, and also termination codon during the translation. So it must carry all these sequences, which must not be there in case of cloning vectors, because we don't need the expression of our gene inside the cloning vector. So if a cloning vector is not having any of this, it won't create any problem. But for an expression vector, it must carry this kind of translational initiation sequences, and as also transcription termination sequences and translational termination codons. Okay. Third important thing for them to have is the element for expression in appropriate host. For example, if we need to express this particular vector inside a bacterial host or a prokaryotic host, it must carry Shine-Dalgarno sequence, which is important, and we all know that. Shine-Dalgarno sequence is important for the recognition and the binding of ribosome and initiation of translation, so it must carry Shine-Dalgarno sequence. Okay. And if we want to express our expression vector in eukaryotic system, it must have the Kozak consensus sequences, right? So it must have the necessary elements for the expression in the appropriate host. Okay. Now the fourth thing is a purification tag. Now most of the expression vector have this purification tag. However, these first three things are the most important part above all, but rest of the part can be present or cannot be. But nowadays most of the expression vector consisting of this purification tag, it simply after our desired gene we just attach several. So we just attach several tag of amino acid sequences. Usually we tag it with histidine sequences. So 6 histidine sequences are used. It is called 6x histidine tag. We are going to see in during the expression vector, we are going to see two different expression vectors, pbad and also pet vector, and in all those vectors we are going to see this 6x histidine tag is present. Why this 6 histidine tag is present? Because this 6 histidine tag is actually helping us to drag our desired product out of a mixture of cell, because what we are doing, everything will be doing inside the bacterial cell. So inside the bacterial cell, not only our gene of interest will express its protein, but also rest of other proteins will be expressed. For example, this blue colored proteins, for example, this red colored proteins, all these proteins and RNAs and DNAs will be extracted. So we need to take our desired protein out of all these things. How can I do that? If we tag it with 6x histidine, it will be easier for drag it out via a procedure we are going to study later. Okay, there is a very important procedure to take our 6 histidine tagged protein out of a mixture of proteins. So it will help us in the purification. That's why it is called the purification tag. And the fifth thing is the fusion protein insert. So you must have fusion proteins or may have fusion proteins like glutathione transferase or maltose binding protein. Now the presence of this type of protein tags right after this gene of interest or our gene of interest or interest of sequence. Okay, now this glutathione transferase or maltose binding proteins, these proteins, what they do actually, they increase the solubility of the protein in the water on the buffer. So solubility of the protein gets increased, which is required in most of the cases. Otherwise we cannot get our desired protein in our hand, right? Or else we can have fluorescent proteins like green fluorescent protein or GFP tagged. Now the importance of GFP tagging is that it can act as a reporter gene, and it can tell us that appropriate expression is done or not. Because if this GFP protein is tagged and our protein is expressed, in those cases what we can get uh, the green fluorescent protein, and we can look at the fluorescence via the fluorescent microscope, and we can look at the expression of the protein in real time. So that's why by tagging it with GFP, it can act as a reporter gene. It can report the expression of that particular gene inside the host cell. So what are the difference between the cloning vector and expression vector? We have already discussed it earlier, that difference in their purpose. So we produce cloning vector for simple cloning and transferring our desired gene into a host cell. It ends its job when it transfers the gene into the host cell. And not only transferring, it ends the job, but it needs to replicate inside the host cell. Replication, up to replication, these cloning vectors work. So if we look at the central dogma of biology, if we are having the DNA, from DNA we are having the RNA, and from RNA we are having the proteins, right? So from DNA to RNA is transcription, from RNA to protein is translation, from DNA to DNA is replication. Now the job for this cloning vector ends in this point of DNA to DNA. So simply cloning vector works up till this DNA level, not to RNA and protein level. But on the other hand, expression vector work of expression vector starts from DNA, and it travels from DNA to RNA. So let me change the color. It starts its expression from DNA to RNA, then RNA to protein. So this work is done by the expression vector. So we utilize expression vector for the expression of a gene, expression of a desired gene, and we use a cloning vector for the transferring our gene of interest into the host cell and replicating it to increase the copy number of our desired gene inside the host cell. Okay, so if we look at the constituents of both cloning vector as well as expression vector, we are going to see the difference. For example, in case of cloning vectors, it consists of only three major important things: origin of replication. If you look at here, you can find this is the origin of replication, and this is an example of cloning vector PBR322. It must have selectable marker genes. To select it, when you plate it in the food or petri plates, it is having the ampicillin resistant gene. That means if we plate this particular bacterial colony onto the plate, onto the agar plate containing ampicillin antibiotic, those colonies will start to grow which are having this gene, because this gene is giving them the ampicillin resistant proteins or ampicillin resistant mechanisms. The third important thing is the multiple cloning site, or we can say the polylinker region. We can see here it is also the multiple cloning site, where we are seeing EcoR1, Hindi3, EcoR5, BAMH1, SAL1. These are the restriction enzymes. It can cleave different regions and segments of this MCS, so that we can enter our insert DNA in between this particular plasmid, right? On the other hand, the expression vector consists of many more things except for these three. So it must have origin of replication, selectable markers, and multiple cloning site. But except for these three things, they also have many more extra things, like an inducible promoter, as we have already discussed. So here it is a promoter for arabinose. So this promoter is inducible. It can be induced due to the presence of uh, arabinose, or presence of the protein arabinose C or ARA-C. So it is inducible in nature. Second thing is that a transcription and translation initiation sequence. So start codon for translation, stop codon for translation, termination sequence for transcription. So it must carry all these things. Elements of transcription for the suitable host. So if we need to express this vector inside the bacterial cell, it must carry our signed Dalgarno sequence, and so on. Now seventh one is the purification tag, or in this case histidine tag. We can see here the express of 6x histidine. So 6 histidine residues, amino acid sequence residues are there, because we require them for the purification, as we have already talked about. And also fusion protein tag. It can be present or it cannot be present. It may present, it may not present. The fusion protein usually can be GFP as a reporter, or glutathione transferase and all these things for proper and better solubility in the water. Okay, so that is the difference between them. The expression vectors are much more complicated than the cloning vectors. Cloning vectors are simpler than the expression vector. Now let us talk about a totally different concept of life cycle of bacteriophage M13. It's a phage particle actually, M13, and why it is important? Because it can give us a single stranded vector. The type of study we have discussed earlier is always the double stranded vector. The vector we choose always is a DNA vector which is double stranded in nature, but we also require single stranded vector in several different cases. What is the use of this single stranded vector? For several applications of cloned DNA, we require the single-stranded DNA. And also for the dideoxy method of DNA sequencing, we actually require single-stranded DNA instead of a double-stranded DNA. So if we use single-stranded vector, we don't need to do much. We just extract the DNA and can go for the dideoxy method of sequencing. And the most important point of using single-stranded vector is that it is very easy to determine the orientation of an insert if we utilize the single-stranded vector. How we can determine the orientation of insert, I'll be telling you later, but we can determine the insert that we are utilizing, what is that orientation. That means if it is orienting from 5 prime to 3 prime this direction, or clockwise or anti-clockwise from 3 prime to 5 prime, like that. Okay, we can derive this mechanism utilizing single stranded vector, right? So how can we get this single stranded vector? The M3 phage particles are such a type of particles that can give us single type of single stranded vectors. Now we can see in this picture, this is a picture of M13 filamentous phage. They are also called filamentous phage. Now in this case, what we can see inside it, we are having one DNA. We are having a DNA which is single stranded in nature, and also it is covered by a capsule which is having a gene 8 protein. It is called the gene 8 protein. Now what we can see, when we transfer this via process, say here it is via the process of conjugation, we transfer this plasmid via the F plus E. coli to the F minus E. coli through the F pillars, and after entering it into the E. coli cell, what is going on inside the E. coli cell? Let's find out. There are three different stages of the replication strategy of the bacteriophage M13 DNA. At the very beginning, the DNA is positive single stranded DNA. Now the single stranded DNA is copied, a new strand is being made from the single stranded DNA via DNA polymerase. So DNA polymerase will act on, it will start making the double stranded DNA from the single stranded DNA, and we soon end up with a double stranded DNA molecule. Now after making the double stranded DNA molecule, this double stranded DNA molecule starts the replication process via rolling circle mode. Now this double stranded molecule or intermediate, double stranded intermediate which is just being made, is also called the replicative form of DNA, and the single stranded one is called the single stranded DNA. So it is called RF form or replicative form of DNA inside the cell. So this RF form will start replicating via the rolling circle mode of replication, as we can see in the stage 2 picture. So it is making it in rolling circle mode. After making it in rolling circle mode, what it can make? It can make a double stranded DNA on its own, like that. Now it will start making this rolling circle mode of replication and making more and more copy of this DNA, but until and unless they reach a signal from a protein called gene 5 protein. Now gene 5 protein always expressed in a very minute amount inside the cell, but sometimes the amount of expression of this G5 protein get increased. Now the process of this double strand synthesis is done right after the infection, right after 1 minute of infection, but the rolling circle mode of replication continues until 20-25 minutes post infection period. Now more than 25 minutes post infection period, when the gene 5 protein is expressed in higher amount inside the cell of this E. coli, in those cases this G5 protein can come and attach to the single strand which is displaced during the rolling circle replication, and it will hold on to the single stranded DNA, and it will make a single stranded DNA ligate a single stranded DNA, and it won't allow this single stranded DNA to be duplicated. So what it does in this case, it is making a single stranded DNA during the rolling cycle mode of replication, and it is not allowing this to be a double stranded DNA anymore. Now this single stranded DNA can be packaged again into a virus particles, and it can come back to its original form like that. Okay, so this is a process of the life cycle of M13 phage. Now it can carry both the type of variety of DNA, single stranded as well as double stranded RF form, right? But we can take the single stranded DNA for our desired purpose for dideoxy mode of sequencing, as well as we can use this single stranded DNA to understand what is the orientation of our insert. Now, okay, suppose we have taken two single stranded DNA. So say this one is a single stranded DNA with our insert, say the insert in this red color. So the insert is suppose in this direction, and say another single stranded DNA, and in this case this one is green, and our insert in this direction. So you're getting the fish hook arrows. This one is in this bottom direction, this one in this top direction. Now how can we know what is the orientation of our insert? We can utilize the single stranded DNA fragments to understand that. How? Say two phage particles are there, so we extract these two phage particles and add them onto a mixture, and we want to look for the production of a double stranded hybrid DNA. How? Because as these DNAs are single stranded, they are having a tendency to be bound with each other if they find complementarity, and we know that we are taking up these DNAs, similar type of DNAs, the same type of DNAs we are taking. So it is possible for them to have a complementary region and to bind with their complementary region. Now if we found that in this case, for example, so what it can happen, say this one, so this will be the case in this case, so this red one and say this green one. Okay, now in this picture what we can see, when we add them in the mixture, we can see they start to form this double stranded DNA. Why they are forming the double stranded DNA? Because the orientation of the genes are in opposite direction. This one is in this direction, this one is in this direction. If both of the genes are orienting in the same direction, hybridization could not occur. So by looking at this double stranded DNA, after providing the single stranded DNAs, we can tell the orientation of those genes in those two species are in opposite direction. So we can get an idea about the orientation of the gene by looking at it. And how can we know there is a double stranded DNA made? In each case, we take this DNA, fragmentized it, and we run the gel, and we can run by running a gel we can tell that whether the double stranded DNA is present or not. Okay, so if these genes in the gene of insert or the inserts are placed in the same orientation, they may not hybridize, so no double stranded DNA hybrid forms. If they are in opposite orientation, then they will be hybridized and a double stranded DNA will form, right? So by looking at it, we can tell the orientation of our insert. So that's why these single stranded vectors are important. Now let us talk about the regulation of the gene expression in pet vectors. This is a kind of expression vector, and we are going to see how complicated an expression vector really is by looking at this picture. Now what we can get here, the most important concept you need to memorize, you need to remember all the time, is that whatever we are dealing with now is regulation of transcription is the most important part. The regulation of transcription of our desired gene is the most important of all. And the regulation of transcription is carried down several different stages to have better regulation, to have better control. The only reason for committing one reaction in multiple small steps is to have a better regulation, a better control over the stage. And in this case, the exact that thing is happening. Now for a transcription to be regulated, we need to regulate the binding of the major transcription enzyme polymerase, RNA polymerase, and also we need to look for the operator and promoter sequences and how they're interacting with the RNA polymerase. And also we need to look for several layers and several different steps of transcription, and different steps are related with each other via on and off switches. So they are working like a molecular switches. Sometimes they are getting on, sometimes they are getting off, right? So these are the switches of that, on and off. So one switch is getting on, it is turning off another switch, and that turning off of that switch, it's turning on another switch. So this is a sequence and chain reactions that is going on. So if you understand this concept, it will be pretty easier for you to understand this whole process, right? Now in this process of pet vector, which is a blending of the vector, as you can see, this is actually the pet vector, and this is a E. coli cell. Inside the E. coli cell, we are having the genome of E. coli, and also we are having the pet vector. But the genome of E. coli is modified, as well as the pet vector is made artificially, and we all know that. The construction of the pet vector is such a way. What we need to know is that during the process of operon, it is having promoter region. The promoter is for lag promoter, or promoter for lag gene, and also operator for the lag gene. Right after that, we are having our target gene or gene of our interest, which I uh, shade it with the green color. And also the polymerase region required here is the polymerase from the bacteriophage T7. So the bacteriophage T7 polymerase, then lag O promoter, then our target gene. And further upstream of all these things, there is a lag I gene to produce the lag I or the lag repressor molecule. Okay, so this is the construction of pet vector, which is an expression vector to be expressed inside the E. coli cell. So it must have that Stein-Dahlgren sequence. It is having necessary all the things like multiple cloning site, origin of replication, origin of transcription, start codon, stop codon for translation, and all these things. On the other hand, the E. coli chromosome is also modified, because it is not a normal type of E. coli host, not a wild type of E. coli host. It is modified E. coli. It is also having a lag promoter, a lag operator, and E. coli polymerase to be sit there. So this is fine. But right after this lag operator and promoter, we insert a gene T7, a T7 gene which is coding the T7 polymerase, right? So the basic principle of this process is that it is a two component system. So the very first component is the E. coli genome and the expression of the E. coli genome. The second part is the expression of the pet vector, right? So the very first thing, the expression of E. coli genome, the expression of this T7 gene is required for the expression of the pet vector or expression of target gene. So there are two genes we need to express, one, and mostly one gene. Actually, this is the target gene we need to express inside this E. coli host cell, right? Because this is the gene of our interest. But to express this target gene inside this E. coli, we must express the T7 gene, because T7 gene will produce the T7 RNA polymerase, which will be required for the expression of our target gene, right? So if we can't make T7 RNA polymerase, so the expression of our target gene will be blocked. So now let's look at the process and the regulation. So finally, the E. coli RNA polymers can come and sit onto the lag promoter region. Then lag operator is blocked by a repressor, which is made by lag I gene further upstream of the E. coli. So it usually come and block this part. But when this lag operator is free of any repressor, in those conditions, RNA polymerase of E. coli can transcribe this T7 gene and can translate this T7 gene into T7 RNA polymerase. So for transcribing and translating and producing this T7 RNA polymerase, we need to take this lag operator, this lag repressor out. How can we take out this lag repressor? By inducing the system with an inducer, and the inducer here is IPTG. So if we provide IPTG in the medium in a higher concentration, the IPTG is going to induce the process, repressor is going to fall away, and E. coli RNA polymerase can sit onto the promoter. It will start transcribing the T7 gene, and it will start translating it, and sooner we get the accumulation of the T7 RNA polymerase inside this E. coli cell. Okay, now what we'll do, this T7 RNA polymerase can go and sit onto the T7 promoter, which is present in the pet vector, But again, the lac I gene is producing lac repressor, which is again bind to the lac operator region there to block the transcription of target gene. Why we require the blocking processes? Because we don't want our target gene to be expressed all the time. We only want to express our target gene when we need it. So we produce in such a way that in normal conditions the expression of our target gene is blocked by these lac repressors. Only when we add the inducers, again here is inducer IPTG is added, and when the IPTG is present, then lac operator is free, lac repressor is falling down. The T7 polymers can sit onto the T7 promoter and it can start transcribing our target gene. Then translation of target gene will happen and desired product will be accumulated inside the yeast cell. So this is a two-way component system, and we are having regulation in both the steps. But another regulation is also there, and further regulation to ensure that not a single time target gene is expressed when you don't require it. Suppose any kind of process, the lac repressor gene is getting blocked by something, or the lac repressor gene is somehow misfunctioning. In those conditions, it may happen the lot of T7 RNA polymers which is being produced now, they can go and sit onto it and target gene expression will start, though we don't want it. To block this action when the lac repressor is failing, we also have another second process of blocking T7 RNA polymerase to sit onto the T7 promoter of PET vector. And the process is that we are producing a gene T7 lysozyme which will hold on to T7 RNA polymerase and block the activity of the T7 RNA polymerase in normal situations. But when we add IPTG again, this T7 T7 lysozyme is getting degraded, it is getting turned off, and the T7 RNA polymerase is getting activated. So to on and off this T7 lysozyme and to express this T7 lysozyme inside the cell, we required another type of small vector containing this T7 lysozyme gene. This vector is called PLYS S vector or PLYS E vector, right? So we require three different vectors: major E. coli genome and two other vectors. One major is a PET vector and a small PLYS or PLYS-E vector for the regulation of the process. So it's a very tight regulation to ensure that only the expression of target gene happens when we desire it, when we want it, right? Okay, so we have seen the expression via the lac operator and lac promoter. Now we can see we can use the tryptophan promoter in similar way. Now before understanding the process of this bad vector or PET vector, you must understand the Operon concept. You must understand lac operon as well as tryptophan operon. Otherwise you cannot do this. You cannot understand this. So I recommend you to go back and look for the video of lac operon as well as tryptophan operon, then come back here and look for this video. Okay, so what we are having here, we are having a promoter region, so promoter of tryptophan, and then we are having the lambda CI repressor. Now this function of the lambda CI repressor is to block this PO promoter which is controlling the expression of our desired gene. So what we need, again we need this desired gene to be expressed only when we require them. So we need to block the action in the normal time, but when we require them we'll add some important ingredients, inducer, which will remove the block and start the transcription of our desired gene. So what is happening in the normal case, when we don't require the expression of our gene of interest, we utilize so we don't provide any tryptophan in the medium. So what will happen, the tryptophan promoter is on, it will start transcribing this lambda CI repressor protein, and as this lambda CI repressor protein is made, it is a repressor of this lambda promoter or PO. So it will come and bind to PO and repressing the activity of PO. So it will block the binding of other proteins into this particular promoter to start the transcription process of desired gene. Now it is blocking it. Then what we'll do, when we add tryptophan, we know that tryptophan operon is a type of operon where adding tryptophan can act as a repressor for a promoter. So it can act as a repressor, right? We all know that. It's an aporepressor. That means normally the repressor is there present inside inside the cell, but it is in inactive form. But when you add tryptophan, so say this red circular thing is a repressor which is in inactive form, but when we are adding this tryptophan, so tryptophan is coming and added here. As a result of this tryptophan binding, this repressor is become activated. So the repressor is getting activated. So now it is in the active form of this repressor, and it can come and bind to the promoter region here. So after binding with tryptophan, it binds to the promoter and it is blocking the activation or expression of this lambda ci repressor. So as there is no lambda ci repressor, nothing is there to sit on to the promoter, so the transcription of our gene of interest can go on and the translation of the gene of interest can go on, so we can get our desired product. So in this case, the inducing property is shown by tryptophan. So just by adding tryptophan, we can reverse the gene expression effect inside the host cell. Okay. Now, regulation of gene expression in PBAD vector. We have seen the regulation using lac operon. We have seen the regulation using tryptophan operon. Now let us see the regulation using arabinose operon. And both of this all of these three different operons are the most important operons to study. So you can find the videos about lac operon, tryptophan operon, as well as this arabinose operon in my YouTube channel. You can go look for it. It will help you to understand the process. Okay. Now again, the basic concept of arabinose operon is that it is also having several regions. It is having operator 1, operator 2, and also inducer 1 inducing 2 sites, and then we are having the promoter for the BAD or the gene BAD coding promoter, or PBAD is the promoter is called the PBAD. And the controlling molecule for the expression of this arabinose operon and for the expression of this PBAD promoter is the ARA-C molecule. Now ARA-C, when it is a single monomer, it does not have any activity, but the dimerized content of ARA-C is a strong regulator of this protein expression. So when the ARA-C molecule is dimerized with itself, it can bind with both the sequence O2 and I1 via this kind of interlinkage and via bending the DNA molecule like that. So it will bend the DNA and attach the O2 and I1 via this dimerized section, where they attach to O2 and I1 via the C-terminal ends of the protein dimer. Okay, so there when these ARA-C dimers sit onto the place like that, protein so promoter for ARA-C is there, so no polymerase can come and sit onto this this promoter region and cannot transcribe the BAD gene, which is required for the synthesis required to be expressed inside the cell. Okay, so this is the particular thing, and what we can utilize here, and what will happen when we add arabinose. So this thing happen due to the absence of arabinose, but when we add arabinose, adding arabinose is dramatically forcing this dimer of ARA-C to be modified. So the conformation is changed for the ARA-C dimer in such a way. It was previously bound with I1 and O2, now it is binding with I1 and I2 instead of IN and O2. So as it is releasing O2, so the DNA strand is again getting linearized from the bending position. So DNA is getting linearized. Now the promoter sequence of this BAD is accessible for the RNA polymerase to sit on and start the transcription. So now it can transcribe the BAD genes and it can give us the products. Now if you utilize this PBAD promoter in the PBAD vector, so inside the PBAD vector we are again having the origin of replication, multiple cloning sites. So these are the multiple cloning site, then stop codon, then also histidine tag. Remember we have talked about this purification tag. So we are having all these things there, but before our desired gene insertion we are having the promoter. So we are trying to express our desired gene under the influence of PBAD, or the promoter for arabinose operon. So we know the funda of promoter of arabinose operon. So when we provide no arabinose, when there is no arabinose on the system, in those cases the expression of our desired gene is blocked due to the dimerization of ARA-C. But when we add arabinose in the medium, it will change the conformation of ara C, and then the RNA pol can sit onto this pBad promoter and can transcribe our desired gene and can translate our desired gene into products. Okay, so providing arabinose is acting as here as the inducer. So arabinose is acting as an inducer again in this case. So in the pBad vector, we are having this particular region pBad, then our desired gene sequence, then the site for enterokinase cleavage, and also the multiple cloning site and all these things, histidine tags and all these things. We are also having selectable marker which is ampicillin here. We are also having origin of replication somewhere here. Sorry. We are also having origin of replication and also having the ara C, because ara C is the most important molecule to control and regulate the expression of this pBad promoter in this case. Okay, now in this picture this is an Western blot which is showing the increase in synthesis of a cloned gene product when the different levels of arabinose is provided to the culture of host cell. Now from the beginning very few amount of arabinose is provided, so the expression of our desired gene is very less. In this first one there is no arabinose provided, so no expression of our desired gene seen. Then the little arabinose, little expression, and as we are increasing the percentage of arabinose inside the medium, the expression of our desired gene is getting increased. So that is telling us that arabinose is acting as a positive regulator or inducer of this pBad vector operon system. And if we look at this structure of this pBad operon closely here in pBad vector, we are having pBad promoter, which is a promoter for the arabinose operon, promoter for the synthesis of gene B, gene A, and gene D. Then we are having the ATG sequence. Then we are having 6 histidine sequences which can act as the purification probe for us. Then we are having epitope sequences. These epitope sequences are aspartate sequence and lysine sequence like that. Right after that we are having the enterokinase sequences which are common to all type of pBad vectors. Now actually there are 3 different variants of the same pBad vector: variant A, variant B, and variant C. Now among all these variants, this up to this part of the segment is constant. So this part of the segment, so pBad, then ATG sequence, then 6 histidine, then epitope, then EK site or enterokinase cleavage site. So up to this part of the sequence is totally conserved in all the 3 variants. But the variable region of these variants are in MCS, because the MCS or multiple cloning site region is different in these 3 different variants. In variants A we are having different like proline, serine, serine. In variant B we are having arginine, tryptophan, glycine, and serine. In variant C we are having arginine, tryptophan, which is similar, but from the glycine it is isoleucine, arginine, proline, and so on. So we are varying in the MCS region in these 3 variants of pBad. Now utilizing these 3 variants uh, we can utilize these variants for our study by cloning different segment of DNAs, different segment of genes for the protein expression analysis. Okay, so that's it, and I hope this video is helping you to understand the expression vectors and their importance. Thank you.